Hi, today I will be talking to you about optical sensing of protein-antibody interactions, our science project. Current advancements in biomedical and biochemical sciences have allowed us to modify the structures of biomolecules, or more specifically in this case, antibodies, so that their functions better suit our needs. After modifications are made, certain tests need to be carried out to ensure that the antibodies function as expected. Traditional methods of testing, such as surface plasmon resonance and ELISA, are surface chemistry methods, which mean that the user has to spend hours or even up to a day carrying out multiple layering and washing steps before results can be obtained. Today's demand for efficiency means that a more quick and efficient method needs to be developed. Today we have here devised a method that only takes about an hour to prepare and 5 minutes for the test. Our assay makes use of gold nanoparticles that have a unique size-dependent colorimetric property. They appear wine red at smaller sizes and purple-blue at larger sizes. Adding salt solution to a colloid of gold nanoparticles produces ions that upset the interparticle electrostatic repulsion and causes them to aggregate, hence changing color. Our assay involves protecting the gold nanoparticle with the antibodies, preventing a color change. Gold nanoparticles were also chosen due to the fact that they do not alter biochemical properties. Now let me take you through the assay. Firstly, a protein is mixed with the gold nanoparticles. A certain amount of salt is added, such that the red colloid turns purple. With this protein conjugated nanoparticles, I allow the corresponding antibodies to bind to the proteins. Theoretically, when I add the same amount of salt that I added before, the steric shielding conferred by the antibodies should prevent the colloid from turning purple. For practical reasons, we will also carry out a negative test using its non-binding antibody to prove that any shielding from aggregation is due to specific protein antibody binding and not non-specific adsorption. And here we have the results. This test was conducted with a 10 kilodalton protein. When the binding antibody was used, the colloid remained red, and with a non-binding antibody it turned purple. We also used UV visible spectroscopy, where a high 650 would mean high aggregation, or a high 525 would mean low aggregation. With the binding antibody, there was a high 525, while the negative test showed a high 650, confirming our results. But when we tried it with a larger 37 kilodalton protein, the results were virtually undistinguishable. Both positive and negative tests showed purple color and had similar 525 and 650 absorbances. Why did this happen? We believe that we have found the answer. According to our source, the smaller protein maintains up to 98% of its structural integrity, hence still having its active site available for specific binding with its corresponding antibody. We also found another paper that discovered gold and sulfur containing groups are very likely to form covalent bonds when placed together. Since our larger protein has sizable number of cysteine groups embedded within its folded structure, we theorized that the formation of gold-sulfur bonds causes the protein to unfold, and hence the entire protein structure is deformed. Specific binding between antigen and antibody cannot happen. Of course, this is just theory, and we didn't manage to use NMR spectroscopy to prove this. In conclusion, this assay would work very well, serving as a quick and easy method for the screening of antigen-antibody pairs and their interactions. Also, the theory that proteins and other biomolecules can still retain their biochemical functionality while being adsorbed on the surface of gold nanoparticles is not true, as we have proven that covalent bonds will and always will present a possibility of protein denaturation. And with this, I end my video. Thank you.